Okay, so I guess we're going to get started. So welcome everybody. Welcome to Judith. Judith has been under the weather recently, not because of the weather, but because of medical problems, and she's here today. So thank you, Judith, for making it,
Yeah. So if you see
dragging
my
yourself
face
out.
disappear, it means I fell off my chair.
And today we're going to be talking about BYOB, bring your own bulb, and we're going to be talking about lily bulbs and scaling. Our first speaker today will be Rick Roddick, and he's from Minnesota, and he's had years and years of experience. He was telling us how he started in high school. I can't get over that. I never did.
Yeah,
Yes. And what was it, 45 years then from high school to now?
yeah, about that.
Oh, you're such a young thing. Anyways, anyways, he has a degree in horticulture, and he spent many years dealing with lilies as well as being mentored by some of the older school, which is excellent. And then we have Judith, and she's had her 50 years plus dealing with lilies on a professional level. And then you'll have me. I won't mention my time. Anyways, so we'll start with Rick. Thank you, Rick, for starting it us off. Oh, and questions are welcome during the presentation. So please, if you have questions, and Judith will be giving some answers as she goes along as best she can. And let's do it. Let's start.
And I will say too that if somebody raises their hand or something, I'm probably not going to notice. So somebody's going to need to pipe in. Okay,
Peggy's good at that.
yeah. So I'm going to talk about the actual propagation of it, and we're going to be talking about what is actually scaling, how to do it, and why we do it and how easy it is, because it really is very easy, but there's a few things that you need to do. Otherwise, it just messes up everything. Next. So we start out with what is a true bulb. All lilies are true bulbs, and it tells us what the anatomy of the bulb is. The true bulbs are actually modified stems and modified leaves. The leaves are the scales. The stem is that really super shortened basal plate. This is different from all the other types of bulbs where their anatomy is way different. You can't take a corm and break it up into lots of little pieces. That just doesn't happen because that's just not the way they're made. Next slide. So when we go to do our scaling, we want to start with a healthy bulb if we can. If people do take bulbs that seem to be not doing so well to revive them, whether they're just going downhill or whether they're actually infected or not, you can do that. You just need to make sure that the scales that you take have all of the disease or infected or damaged parts out of them, so that they'll be able to go on and produce a new plant. Of course, with viruses, though, this doesn't matter because the virus is everywhere in the bulb, so you can't really just take parts of and not get the virus.
Can I
So cleaning,
can I add something, Rick? A lot
yeah,
of times that's an indicator that'll tell you if it really is a virus or something else. Because if you scale it and you don't get a nice little crop of bulblets, or they're really, really slow or tiny, often that's because it is virus and it's not your fault. It's just that's one of the characteristics.
oh sure, yeah, and also we have. I could almost guarantee every one of us has virus lilies in their garden. Um, and it doesn't necessarily mean that they're not going to grow fine for you.
Well, how many people have not viruses in their body? Everybody has viruses. It's
There
part of
you
the
go.
package. Okay.
That's right. That's exactly right. So when I talk about a clean bulb, I don't mean that you've got to have it spick and span. You just get the big chunks of dirt off of there, so that you have a little less probability of introducing bad things into your system. I don't recommend washing them unless you're going to use a fungicide. I generally don't use any fungicides just because I'm lazy, I guess you could say. Also find that I don't really need it if I watch what I'm doing, and we'll learn about that too. Next. So the scaling basics is you start with the bulb and you break scales off, or you cut them off, and you end up with the scales that you put in some kind of a propagation environment, the medium with the right temperature and moisture and stuff, and you get new plants. And of course, those new plants are going to be clones. They're going to be the exact copies of the mother bulb. Next. So we'll talk a little bit more about the basal plate here. Um, you can see in the diagram on the bottom right there, it's kind of triangular formed in most Asiatics. When I first learned about them, I kind of assumed, oh, it's kind of like a dinner plate, but no, not at all. As far as I know, there are no flat basal plates. And that's just because of the anatomy of the bulb. That's the way they grow. You will find differences in different species. For instance, mardigans have a very tall triangle shape for their basal plates. Asiatics have lower ones. I don't even know what orientals or what some of the other ones are. But the idea here is that you want to try to get to break the scales as close to that basal plate as you can. For a few different reasons. One is that, the closer you get to the basal plate, the bigger the scale you have. And the bigger the scale you have to start out with, the bigger the little bulblets are that will be able to grow as you scale them. The other reason is that for whatever reason, scales will tend to be able to produce little bulblets a little easier the closer they are to that basal plate. So it's not like if you break it off and if you break off the outer 2/3, it will never produce scales. But if you took that same scale and broke it off at the basal plate, you might get 5 nice big bulblets. But if you took that 2/3 size one, you might get 1 or 2. I don't know. And it makes a big difference on the cultivars and the types of lilies that you're using too. There's a lot of variation in your success ratios. Next. Okay, so when you take the bulb scales, of course, the biggest scales are going to be on the outside of the bulb. And the biggest ones, because of where they are, they always have a really strong curl that goes around and under the bulb to attach to the basal plate. And this makes it really hard, actually, to be able to break it off really close to that basal plate. So when you're doing that, what I'm saying is when you break them off, you're not going to go willy-nilly and snap them off. You're actually going to go slowly with your fingers, kind of move it around and figure out how the best way is to break that off so you can get as big a scale as you can. And that can be different from bulb to bulb too. Also, I talked about this curling. If you were going to scale the whole bulb, for instance, as you work in from the outside part, that curling becomes less and less to nothing, actually, so that the breaking off the scales becomes easier and easier, but then the scales get smaller and smaller too. Okay, so you break them off. I like to break them off myself, but sometimes if they just doesn't look like looks like they'll break where I want them to, then I might cut them off, or I might just start a cut so that I can finish breaking them off. I prefer breaking myself just because when you do the natural breakage, you don't cut through as many cells, and you get less of that cell insides spreading all over the place that might cause some encouragement of diseases later on. So you have them broken off, and now you need to let that cut surface dry well. At least a few hours. I like to go overnight or even a day, and make sure that cut side is up, not pointing down, because if you have it pointing down on a paper towel, for instance, that paper towel is actually going to absorb some of that weeping material that is being exuded through the cut surface. So just like in a tree that you cut, it exudes a sappy material that kind of hardens up and helps to produce a bandage. Um, and if you don't have any of that matter to produce the bandage isn't there, or at least it isn't there as much. So always let them dry surface up and you'll have the best healing. Next. Now the media, this is another really, really important thing. What you use is really not important, but except that it needs to be able to you need to be able to regulate how much moisture is in there. If you get it too wet, you have to be able to get it to dry out in a relatively quick manner. So that soil is usually not a good thing to use for that reason, and for other reasons, like soils will always have other kinds of microflora in there, soil flora, whether it's diseases or good microorganisms or bacteria, whatever. You want to just try to avoid all that stuff as much as you can. And this is one of the important things that I do, why I don't need a fungicide. So there's lots of different things you can use. You notice that three of those four different kinds are actually made of rock. You've got the sand, you've got the perlite, which is a puffed. Actually, perlite is puffed perlite, and vermiculite is puffed mica rock. The sphagnum moss is good to use because it's virtually inert in that it has very, very little nutrients in it. And you don't want any kinds of nutrients, extra nutrients in your media. All the food that you need is in that scale. The whole purpose of your scaling is to have that scale transfer all its food into new little bulblets that will turn into new little plants for you. There's no need for giving it any extra nutrients. They just need the right amount of moisture and the right temperature. So that, technically your media could be air, I suppose, if you had the right other things with it. Usually it's just better to have a little something else with it, because air is really super hard to control the amount of moisture that's in there. It varies a lot according to just a couple degrees of temperature. And it doesn't hold much to begin with. So if it loses a tiny, tiny bit, that amount of moisture that's lost is a large amount compared to the whole that's in there. So having some kind of a media in there to give you a large amount of holding capacity for the right amount of moisture is important. Next. So on the left there, you've got the raw sphagnum moss and the milled sphagnum moss. Then the other thing, of course, is just the peat moss that people use to mix in their potting soils, for instance. You can use any of those. I have my friend who passed away, Lauren Odegaard. He used the milled sphagnum moss with sand, and that's fine. You could use milled sphagnum moss with perlite. You could use it all alone. You just whatever you need. But the good thing about the sphagnum moss is that right at that juncture of where there's just the right amount of moisture, and where there's not enough moisture, sphagnum moss will actually change color. It'll turn a lighter color when it dries out too much. And the easiest way to tell is when you use the raw sphagnum moss, which is there on the left. The milled sphagnum moss would be next easiest, but not as easy. Next.
Oh, wait a minute,
Oh, yeah.
can you go back for a second? Because I wanted to tell you my prejudice is
Oh,
always
good.
for a really coarse vermiculite, and it's very hard to find it coarse enough because what you're trying to do is make sure they have enough moisture, but not too much, and they have enough air. And it's the pore space that makes some of these things difficult. That's why I don't like to use peat moss. Plus, peat moss really does carry a lot of fungus, gnat larvae, and things like that will get in. If I have to use peat moss, I run it through my pressure cooker. I put it in a turkey bag with moisture in there, and I run it through there first, to really kill it.
That's cool.
But then I mix it with something else. But the way you can tell intuitively is if you have a bag of whatever you're going to put it in, could you stick your head in that bag, seal the bag, and could you still breathe? Could you get enough air to breathe? And that tells you if you've got too much water, that's not going to work. If there's no pore space, that's not going to work. So I think your point is really good about the raw sphagnum, that's not just the color, but it's coarse enough. There's air in there. And with the vermiculite, it's really hard. Though the small stuff is not the best. You really need the chunks that they used to use for insulation. And the way we used to prepare it, because I've had as much as 8 acres of scales, we would do them in big trays, and we would we would just fill up the tray about half full with the coarse vermiculite, just soak it down with water with a hose, and then let it drip. We'd put it in trays with big holes, let it drip for several days until no more water dripped out. And then it's holding a lot of water, but it's coarse enough, it's holding a lot of air too. So the points that you've made are really good. Yeah.
Okay.
Yeah, I
Excuse
would
me.
use
I
the,
have one question
yeah.
from the chat. Can you tell that a bulb has virus by examining it only, or do you need to see it from when the flowers, Judith?
Oh, it depends. There are some viruses that you can see. You're probably, nobody here is old enough to remember brown ring virus, they called it. And it was in the purple bulbs. You'd see the brown rings on the scales and it was from a combination of viruses, but there's so many different ones. You'll know there's something wrong by that they won't scale well, but you can't, for most characteristics, you can't see it. For lily symptoms, you can't look at it and see. But you'll notice
The
that,
bulb doesn't look quite right.
yeah, but there are some in which if they have the red pigment in the scales, you'll see white circles, and that is for sure an indicator it has tulip breaking virus as well as lily symptomless virus. So
Okay, Rick, sorry I disturbed you.
Interesting. Yeah. Yeah. So I was just going to say, yeah, the coarse vermiculite is what you would always use if you were going to go the vermiculite route with the scales. I generally go with the finer vermiculite if I'm doing seeds in little baggies.
yeah,
Okay, next.
it's really hard to get the coarse stuff in
It is now, yes. So here I've got some ready to go in bags of perlite, and the brown that you see is the bits of raw sphagnum moss, and down at the bottom in the middle one there you can actually see a scale peeking out, and that's how I do it. Then what you want to do is you want to have it in a warm, even temperatured place. It's very important that it's even temperatured because you don't want that fluctuation in temperature, because then you get condensation. Condensation is a bad thing. Free water is a bad thing because that encourages disease spores to germinate. And remember, the scale has everything in it, doesn't even need any extra moisture, but you can't be losing moisture either. So everything is in there. You don't need to give it all kinds of extra water to get it to grow. You just need it to be able to sit there and do what it's supposed to do. And then the other thing, of course, is for a lot of lilies, it's very important to keep them in the dark. These are especially are the ones that will that don't need vernalization, and we'll talk more about that a little later, where they can start sending up a leaf even before they've finished growing their bulb. And this is not what you want. You want that scale to produce a nice big bulb, as big as it'll go, before you're ready to go out there and plant it. So a nice dark place, a nice even temperatured place. The picture there I've got in between my towels, because the linen closet for me is in the center of the house upstairs. It's in the middle of the house, in a little closet that the door that stays closed the most. So that's the most even temperature I have. So, whether it's 70 degrees or whether it's 65, probably not a big deal, except for it'll take longer at 65 than 70. And it probably would be noticeable too. Next. Oh, and one more thing.
I
Yeah.
used to stick mine when I was first doing just fun things for myself. I would just stick them in my laundry room in a tray, and then I would put like a black bag on top so they wouldn't get the light. That's another way to do it. And then the other is, if you want to be worried about the condensation, we always don't have anything that's completely tightly sealed. We punch that's what the media is, to hold enough water to correct for that. And you can punch the little tiny holes in with a ballpoint pen or something. And when I did it, when we had all the scales in great big trays, layered like a pizza, then the liners, the plastic liner that we used in there always had big holes, big air holes. And then when you folded that over in the middle, I should have a picture to show you, you left a space big enough to stick your hand in. So that's why we needed a lot of vermiculite. But that vermiculite is what would carry me forth when I would have to plant that out like the following May. And it would always be hot by then. You're waiting for the fields to dry up. But that coarse vermiculite, you just plant it right in the ground with your stuff, and that would carry you through. So, but the temperatures, and then sometimes I used to stick them under my bed. It just needs to be, for me, my rule of thumb is I always want to do what's the easiest way that's going to work. And for me, the easiest way is to scale everything and let it, I used to have all those trays of scales in a little tiny room with an oil filled radiator. That's what I did later commercially. But I would just let them keep forming their little, roots and root hairs and bulblets and all that until by Valentine's Day. That's when everything goes into a cooler. And then you just let it sit there, or like your garage, just let it sit there till you can finally plant them. So that you don't need to worry that you got to have it super, super warm to get a lot of early development, because you're going to have all winter long before you can plant it out anyway. And that'll give you plenty of time. The scales usually for Asiatics, 2 months is usually long enough that you'll be able to tell when the bulbs are not getting any bigger. And then, they typically, but it won't do any harm if they're 4 months, as long as they don't dry out. And then it's the same thing. They need like between 6 weeks or so at least for the Asiatics vernalization cool period. And they can go 12 to 16 weeks is no problem. They'll just sit there until you finally plant them out. So it's not like a hard and fast thing that you have to really worry about.
I have another question from chat. And the question from chat was, are there any special techniques necessary to scale stoloniferous bulbs, American species?
Well, I mean, it's still, you've got to get, the principle in scaling is you want the biggest bulb you can get, and you want the biggest scale you can get. So the only problem with those is it's still got to be not little teeny bits. You want the biggest bits you start with, and it's got to have a basal. The equivalent of the basal plate is is the equivalent of the meristem. So,
Would that be on the mother bulb then?
The new bulb is probably the better part.
Okay.
And I was going to say, when you're scaling the bulb, you'll start scaling from the outside in. And when you come to this last year's stem, it's really interesting. When you reach that point, that's the part that developed over the last growing season. And if you keep going, you get to these little tiny scales, and finally you can see in there a little nubby of where the next part is, going to be. So you can use that inner stuff for tissue culture, but it's not worth scaling. You might as well just drop the little bitty part inside, the little teeny part, just drop it in and plant it like a tiny bulb. So I haven't done a lot of the stoloniferous, so I just, and a lot of times people use the term stoloniferous and stoloniform to mean the same thing, and they're not the same thing. And that's,
That's right, yeah.
yeah, and so you have
Stoloniferous,
to clarify, do you mean the bulb itself is formed with the stolon, or do you mean you had an underground stem wandering that formed little bulblets all along the way? And so that's not the same thing.
Okay.
yeah, that's right. So yes, though the stoloniferous bulbs like Pardolinum or Superbum like Michiganense, um, the basal plate is that whole stem part. But as Judith was saying, the closer you get to the end of it, the better it will be. But then there'll be always that trade-off of big scales versus small scales.
Big piece, yeah. And that's one of the problems in scaling a lot of the new things that are polyploid. And people think, oh, it's so great because they're so gigantic. But the problem is they have to be gigantic for you to ever be able to scale them. I mean, I had Shaharas on for probably 10 years before, and I thought it was a terrible lily because it wouldn't scale well, because I didn't realize it was a triploid and I had to have gigantic bulbs to scale. And so that, you learn some of those things as well. And it's genetic as to whether, like a trumpet lily is not ever going to make as many scale bulblets as an Asiatic will, because they have really strong apical dominance, and the first scale bulblets, one or two that form, suppress the formation of more of them. But an Asiatic will go on and make lots, but they'll be smaller. So it's kind of inborn. And when I do tissue culture, I always notice that, because it's completely the same thing. The ones that are easy and make lots of tissue cultures will also be easy scales. So, yeah.
Mhm.
Okay, Rick, continue.
Okay, so yeah, so there's not just one way to do scaling, but as Judith was saying, her way is a little different, is going to be a little different than my way, but the important thing is still no water condensation in there. We don't want that extra water laying around. Next. So with my method, I go, and when I find that I have the right amount of moisture in there, the little baggies, there are no little baggies that hold all the water in. They'll hold liquid water, but they will not hold the water vapor completely in. They're somewhat permeable that way. And especially since with scales, it's not just a couple weeks that we're doing that with, it's going to be a couple months or more. You need to take that into consideration. So what I usually end up doing is I'll put them all into one big freezer bag, which is pretty impervious to vapor. That's the whole purpose, to have a freezer bag. Next. So hopefully this is going to be what you're going to end up with, is some good scale bulblets coming. There will be lots of variation in them. These were all scaled and put in the bags at the same time, and you can see that the Orient Pets up there went a lot quicker than the Mardigans below. In fact, you can see some of the Mardigans have not even started yet. In fact, I think if I remember right, I was a couple of them that never did at all. But that's the way it is sometimes. You can also see down at the bottom there with the Mardigans that, I'm not the best person to break them off either. But it is what it is. Next.
Oh, Rick, I was going to tell you, I thought you would find this funny. In Holland, they use a machine that for the people who scale it, and it's just metal stick, and they just wham the bulb on top of the stick, and then it rotates like mad and flings the scales off. So can you imagine how much damage and loss you have? You know, and of course they
Wow.
don't care because they just are going to use it by the ton practically. But
Yeah.
it just made my stomach hurt to see that, to think of all the unnecessary damage. But your pictures are just wonderful because you can really see the moisture level and air level were both perfect, that you got little root hairs that everything's stuck onto. You're
Yeah.
just fantastic.
Yeah, so that's what this is showing also. It's got the perlite again, and the perlite is sticking to the roots because that central root, that is the linear part, has all kinds of little hairs around it. And that's what's making the perlite
Yeah.
stick to the root. It's not just the root, the major root itself. So when you go to plant these, you don't try and get rid of that perlite. It's not necessary, and having that perlite in there is actually a good thing because it will allow for a little better air circulation temporarily, at least in the soil that it's in. That will help it along. But if you were to try to take them off, you're going to be stripping off all those little root hairs. It's not at the end of the world if that happens. Root hairs grow comparatively, they grow instantaneously in just a day or two. But it's still not a good thing to provide more damage than you need to because it's always a possibility of further infections. Next. Okay, so here we have another mardigan there ready to go and ready for vernalization. The bottom right one is ready to go for vernalization. Vernalization is when you take any plant that needs to have some kind of environmental cue to make it go to the next stage. And for some things, it might be growing roots. For most things, it's growing top growth. And for something like a Mardigan lily, they have to go through a cold temperature regime to get it to have special chemical changes inside the bulb to produce a leaf or leaves. Asiatics all need vernalization. All the Mardigans do. Some of the in general, most of them that are in the northern parts are going to need more vernalization than the southern parts that have the where the species parentage are from. Next. So here we have some more here, and remember, Judith was talking about those big scales. Those big scales are going to produce the big bulbs, and with Orient Pets, she was telling me that if you start with big scales, you can probably get a scale bulblet that you plant in the spring and get it to bloom in the fall.
Yeah,
And that's
that's my,
pretty
and
amazing.
for lots of scales, that's how I tell I had the right size. If half of them produce not just leaves, but a little tiny stem, then you did exactly the best size. And I think I'm the only person who ever, well, I think I'm the only other person who counts chromosomes and looks at all that stuff, but I want to know, if it means that you don't have to grow it a third or fourth year, it's worth to know that. Yeah.
Mhm.
This is really beautiful because you can see in the top picture, you can see where it broke off, along the basal plate. And you'll see that form, you'll see that's where a little ring, a little row of callus cells will form, and then the bulblets come out from there. Yeah.
Mhm. So the trumpets won't need a fertilization, so it's going to be very important that you keep them in the dark until you're ready to go. Orient Pets, because they have the Oriental parentage in it, they probably would want some fertilization to get going.
Okay, question from me. Is vernalization would it be helpful for the trumpets?
It's
Judith?
it's just kind of, it doesn't harm them as long as you don't have them get exposed to the light. And that's the problem when you've got trays that you're going to go in there 2 or 3 times through during the season and look at them. If you turn the lights on when you go in the room every time, all the ones on the top will have sprouted, and they'll
Okay.
have these long straggly leaves. But they'll, they'll probably, if you kept it cold in, they'll probably still make some new ones, but I don't think it'll necessarily do them harm. It's what it is that the light triggers the same things that the cold would trigger. And
Okay.
it's the same thing like with hypogeal seed, with oriental hypogeal seed. You can, if you expose the seed, the little bulblet to the light, once the bulblet is formed, it doesn't have to be fertilized then. You can cheat, but you only do that if you've got a handful of things, not if you've got 8 acres full of stuff. So
Yeah,
Okay.
and of course that's not necessarily for all the lilies.
no, no, no,
But
no, no, no.
she's absolutely right that the light can take the place of cold vernalization for some of them.
Yeah.
But the cold added to a scale bulblet that does not need fertilization, it doesn't do anything bad. Unless it doesn't
It
like
doesn't help in any way. It's just another way to hold them. That's
Yeah.
it. A lot of times you're doing it just to make it practical so that they'll hold until you're ready to plant. You till the spring rains are finally finished in the end of June sometimes, and you just got to hold them a long time, and so that's a different matter. Yeah.
Yeah.
And I believe that the trumpets, what they need to go into the next cycle of growth is moisture. I think their physiology carries them through drought, and when the rains come again, that's when they are ready to leaf out and do their thing. But that's just my prejudice, and I haven't ever done anything to prove it. But I know when I would have them stacked up in my barn, which had no heating whatsoever, all these trays, the trays that were on the bottom where they would not dry out very much, they would be damper. Those always sprouted earliest. So that's why I have that prejudice, but it could also just be happenstance. But oh, your pictures are so good. I just have to keep
Thank
oogling
you.
your pictures.
Well, this is the last slide. So now if there's any other questions that
Why don't you switch to that slide? Yes, thanks.
Yeah.
Any other
If there's
questions?
any other things that people might have something about, let's see if I can say.
How long fertilization? How long a period?
Well, that can depend too. On Mardigans are probably will want the longest vernalization period. And if any of you have talked with Don Crook, he will tell you, as most people will, is 3 months is the general one. But if you can leave it go for 4 months, it'll be better yet.
Yeah, and it doesn't harm them to do it longer as long as they don't dry out. And so generally, it just depends. You say, well, when do you want to plant them in the spring? And you stick them in there around Valentine's Day, and where it's cooler, or out in your garage or someplace, but where they won't freeze, but they'll, but as a general rule of thumb, it's just what you said. Like the Asiatics usually don't need more than 6 weeks, but you're not going to go out and plant in the middle of March in most cases. I used to be able to do that, but not anymore. But we just hold them until the soil can be worked in the spring, and by then everybody's had enough cold. So
Because it's too wet in
well,
March
yeah,
now.
12 to 16 weeks is long enough for anything. So
Rick, I have a question about adding moisture to your growing medium that you're putting your scales in. Do you put a little bit of water in or not?
So, well, I moisten everything up before I do anything, and I usually start with a bowl of the perlite, and I'll put water. Oh, and by the way, I usually use um um distilled water, just because I'm a picky person, and I'm thinking that it's gonna introduce less possibility of diseases or bad things in the medium that I want to keep semi-sterile. Um, So anyway, so yeah, I'll start in a bowl and mix up a big batch. And sometimes, I might have to I always leave things be there for a day because it's kind of weird, but the moisture, the water will seep into the cracks of even the perlite. The perlite surface is actually not smooth. It's very rough. Um, and the moisture will actually creep into all the little cracks and stuff overnight, just like they would if you were using vermiculite. You've got those plates, and all in between the plates there is dry until you start filling it up with water. So you can put water in your perlite or whatever your medium you have in the evening, and you look at it and you say, oh, it's too wet. By next morning, it's just perfect, or it seems to be at least. And if it's not, then you just want to add a little bit more dry medium to it. Um, it's always a little easier to add more moisture than to subtract it once it's in the baggie. But if there is too much condensation, then you're going to open the bag. You're going to want to hold it open somehow. Lauren always used a cotton ball because it still had lots of air porosity for transfer through, and it actually absorbs some moisture also at the same time. Um, and then, after a day or whatever you think is right, you'll want to go back to whatever you had, whether it was with me where I would close it up completely, or what Judith says, keep it open a little bit. Then you're going to want to be testing that out for the next day or two and see again, is the moisture capacity correct or not? And then you're going to need to either subtract more moisture or maybe add a little bit. But if you add a little bit, you not very much, what, maybe, I don't know, 10 drops. That's what I would start with.
And Steve had a question.
Yeah, thanks, Brian. Excellent slides, Rick. Do you have any? So I have two questions effectively. One is, would you recommend practicing on something that's a bit more expendable than trying it on your sort of, favorite hybrid? And do you recommend lifting the bulb completely to break the scales off, or can you sort of dig down around the side of the bulb to expose part of it? Does it have to come out of the ground completely?
Yeah, there's a lot of people who have done that, where they've dug down and left the bulb there intact. I've never done it myself. It just seems like a lot of work.
I can never do that without damaging the bulb and getting a crushed scale. So I always, lift it. And the other thing, Peggy, I wanted to say is when you're talking about, how moist is your media, if I'm just doing a little bit of vermiculite for like one bulb, a lot of times I'll just mix it up and add the water just like Rick does. And then I'll just let it stand in a colander, like draining your pasta, and just leave it in the sink for a day or two. Because vermiculite holds a lot more water than perlite does.
Yes, yes, it does.
A lot more. And that's why it needs to be so coarse, because it means you also have to have more airspace to make up for that. And so you got to let it drip longer. Yeah.
And there was a question about whether vermiculite
Yeah.
or perlite. Is there any difference in the media or?
Oh, we'll finish with Steve first.
Okay.
The other thing about digging down for the bulb to break the scales off, well, then you're going to be putting soil back in, and that soil is not having that air capacity again. So you do have to, if it were me, if I went down to dig it in there, I would leave that hole open for a day before I filled it back up. And what was the other part of your question again?
Steve. Yeah.
No, I think you've answered it. I was just thinking that I should practice on some
Oh, yeah.
Asiatic bulbs before digging up my sort of favorite, Aurelian hybrid and trying to scale that.
Yep. Sorry. Hi, Steve. You were asking about the American hybrids and the rhizomatous stoloniferous bulbs, and I made trials about these, and what I found is it is best to treat the American hybrids and species like a martagon. So you have to keep it 3 months at about room temperature and give them at least 3 months of cold. What I've found in my trial, and these are preliminary results, that if the bulblet is at least 20% of the size of the scale, so it's like in length, it is time to start cooling them. That at least that's what I found is best. And I also was fortunate enough to get bulb mites, and it is best to roll the scales in sulfur powder because it will kill the mites. So if you use mulch, for example, straw as a mulch, there are also straw mites which behave like bulb mites, and they start eating your bulbs and scales and bulblets. And I rolled them in these sulfur and then put the scales in the bags, and they were fine and healthy.
So, and as far as the practicing part, I mean, I did my first time when I was still in high school. Actually, it was just to try it to see if it worked, and it was just with a hybrid, and it's surprisingly how easy it did work. And I used perlite way back then too. But I didn't know about using sphagnum moss, so I just had it plain that way. So I was pretty lucky. But on the other hand, I don't know if I was lucky or maybe I just had a really good mentor, because I had two friends, actually, they were friends of my parents at the time, that were members of the North Star Lily Society back in the 70s. So they could coach me along. But I don't you might want to just try an easy one just to see first. But I guess it wouldn't, I would, if I had a reason why I needed to do it, like if the certain specialty bulb was going downhill, I would probably do it. I would probably do it anyways.
Oh, I wanted to ask you something, Rick, that I have never quite figured out, that I'm still trying to figure out. And that is the peculiar performance of Henry eye. And you see that also like in Black Beauty that has a lot of Henry eye. You'll scale these gargantuan bulbs and then you put them in a nice, moist, wet, warm place. And 3 months later, zero, nothing has happened. And it seems to be like if you start to do it a little too late, if you did it a little earlier, they would go over the process. And I have done this for several years, that we would get nothing to happen. And then we would finally put it into the cooler, these lumpy looking things. And over the summer, finally a little bit of something would happen. And so it would be like a year later, they would really do their thing. And I don't know why. Henry, I don't
Yeah,
know if it's a function that it was too big a bulb or if we scaled it too late. I don't know what's going on, but that's one of the cases when it's what you're talking about. Don't be afraid to plant the media along with the scale, because that'll keep it going. Even if it never made any roots or anything, it's just sitting there in the ground, and then in another year, suddenly it'll all work.
that is right too. I've never scaled Henry eye tights before, actually. Never had the need to. But I will say that I remember reading years ago that the Caucasian type lilies in general, I read somewhere, some scientific paper, that they came up with the scaling was actually best done in the summertime after they bloom.
Oh, so I wonder about
Now,
that. And like Black Beauty is the other one, because we used to scale a lot of it, and I learned to not wait too late in the fall. Most of them, if it doesn't matter, I mean, you've got all those months until you're going to plant it next May when the ground finally gets dry. So you're not in a hurry. But I know if I waited too late, also speciosum would do that sometimes. So
hmm.
there's still things I want to figure out. There's still fun things to learn.
Well,
Yeah.
isn't that interesting? Because Black Beauty, you know how they get those nice big fat buds with nothing in them too,
Yeah, yeah.
or pods, I should say,
Yeah, I know
seed
I
pods.
know exactly what you mean. I can feel it with my fingers. Yeah.
Okay,
Very good.
I guess we're moving on. Okay, Lynn. We're into bulb color now, Lynn. She
Uh-oh,
with us or not? Let's see.
there we go.
Okay. Okay. Where are we? Bulb color. What does it tell us? Oh, we're doing something. Okay. Okay. Bulb color. What does it tell us? Uh, Bulb color does tell us a lot, especially with the new hybrids coming out. Lily bulblets and bulbils come in different colors, and so do lily bulbs. Next slide. In each division of lilies, the bulb has a different color. Asiatics, the bulbs are basically white. Martigons, the bulb color varies from light yellow or a greenish yellow, with the odd ones with a touch of red. Divisions 3 to 5 are similar to Martigons in color. Division 6 has that classic Indian red color, such as Henryi and the trumpets. There's always that stronger color red in the scales and the bulb with a touch of yellow. Next slide.
And the red bulbs, like the red trumpety things, it's like the difference between a red onion and a yellow onion, you know. So it's,
Yes.
and the other thing that can affect it, there are things that affect, so you'll dig the bulbs at first, things like tiger babies, and you won't see so much of the red color, but if you leave that with a lot of sun on it for a day or two, they'll get a dark glossy purple, so,
Yeah.
you know.
But
And
there's
also,
always a touch of red when you first dig them up if they're a true
yeah,
hybrid.
there
I'll
will
go
be
over that.
some.
I'll
And
go
but
over
you
that.
can also get a little bit of that with hybrids from Davidii, and I don't know why that is.
Oh, okay. I didn't know that
It
part.
may be because we've always selected for the darkest red hybrids. I don't know. But
Okay.
so
Bulb color
don't, just because
reflects
you
their
didn't see the glossy red in the beginning. And that's what we're always talking about, trying to figure out if these things are true trumpet Asiatic hybrids. If they really are, you look at bulbs of tiger babies that have been out of the ground a day or two, and they'll get quite purpley, you know, which you certainly would never get if they were just pure Asiatics.
So the bulb color reflects their ancestry. The color depends on the species which dominates, and the color of the bulb will indicate whether you have a true interdivisional, as Judith mentioned. Next slide.
And I would say not just the color, but the form. Look at the form of
Oh, we're going into that.
yeah.
And the form. Okay, Asiatics, they usually are white, upper left, but if you expose them to the sun for quite a while, after a week or two, they will turn reddish, but it's over a period of time. Now, the trumpets, they'll turn reddish quite quickly, not as, but the Asiatics will turn that sort of pinky red where the trumpets will have that Indian red color. Martigons have that yellow with the white. And this is the mahogany bells. It's one of the few Martigons that has a touch of purple in it. Maybe it's because of the ancestry.
The other thing,
Singagensi,
like where we are, where it's too wet in the winter for the martigans to be truly happy, they'll get yellow just from being sitting in water over the winter.
okay. And then we have, there's another Mardigon here that's more darker colored yellow, depending on the cultivar. Next slide. Division 4, your Candidum, you see there's white and yellow, just like your Easter lily. I couldn't find a bulb of a picture of an Easter lily bulb. And if anybody's had Easter lily bulbs, you know they're yellow and white. And Division 6, this is Henryi. As you can see, you've got that Indian red color at the top. The bottom is yellowy. As the bulb matures, it'll turn all dark red, really dark, dark red. Just like the picture of the scale of pink jazz, that's the color the bulbs will turn when exposed to light. Next slide.
And it's the color of a red onion. That's
And the
what
color of a red onion.
yeah.
Well, it's Indian red. So here's again, top pictures, Henryi, bottom pictures, Lilium sulphurum. And you can see the scales. There's that little bit of yellow that will disappear with light, and all the rest will, like, especially the sulphurum. You see how dark that scale, it will, the whole bulb will become like that. Next slide. And here's species bulbs again, just to let you know. Next slide. Now, Orientals, they can vary. Some of them will be they have that touch of yellowness to them, but some of them are very white, like Dianthia was white, whereas Siberia was this is more of the one on the left, Siberia, the Oriental. That's more of a classic color of an Oriental bulb. Next slide. And then we have the OTs and the interdivisionals. You'll see here there are three different types. One's Corvette, one's Redford, and one's Zambezi. See how the color varies. And I think that's due more to the ancestry. It does not depend, bulb color is not dependent on flower color. It's more what's making up the ancestry of the bulb. Next slide. And here's some other interdivisionals. These are LAs. And LAs will be more white, but you will find a touch of yellowness to them in a lot of the LAs, just a tiny bit. Anything else?
LAs are scale really easily too.
Well, they multiply easily. That's the one thing about them. Okay, next slide. Now we're talking interdivisionals with complex background. African lady was a cross between Orientals and Nepalense. I guess it was back crossed again with an Oriental. Here you see, though, the bulb. This has been exposed to light. But from what I can remember of the Nepalense that I have seen, Judith, have you seen Nepalense bulbs?
Not for a long time. I was just trying to picture them in my mind. I've
They're
got pictures somewhere,
they're
but that
dark
doesn't
like
really look so much
this.
like Nepalense either. I mean, that looks more trumpety to me than.
Yeah, well, its color is what I remember of the Nepalense bulb. The color is dark like this, but that's, see, now we have the oriental shape of the bulb more
Yeah.
so than the Nepalense. The Nepalense is more teardrop
Yeah, I
shaped.
do remember that. Yeah.
Yeah.
Mhm.
So here, there's the picture of the flower and the bulb. So we know the bulb is telling us it's a true interdivisional cross because it's got mixed characteristics from both parents. Next
And the other
slide.
ways you can tell, very, very often, you can tell by sniffing them, because a lot of the fragrances are not, they're just suppressed. And so like if it's a trumpet Asiatic, even 2 generations later, if you've got a whole bunch of them and you stick your nose there, you'll smell the trumpet smell. It's diluted. And so I'd be very curious, because Nepalense has such a distinct fragrance, if you pick that up or not, so
Yeah.
that you look at the form, you look at the bulb form, you look at the flowering time. Is it intermediate between the 2 parents or not? And if it's not, it may or may not be a cross, so you look at all those different things. And so the bulb is a good place to start.
Okay, here's fusion. And there's a picture of the bulb of fusion on the left, and there's a picture of the flower on the right. Now, fusion, that looks like sort of a combination of the bulb
Yeah.
of Easter lily and Pardalinum.
Yeah.
I,
And usually if something is a true cross and the parents are very distinctly different, you will see the traits of both parents. It won't be an either or, you'll see a blend.
both, and that's what this looks like. It's a
Yeah,
blend of both.
That's really a good illustration.
Right. Whether it's a Pardalinum cross Longiflorum or a Longiflorum cross Pardalinum, I don't know. But as you can see, fusion is a classic example, being a true cross where you have characteristics, a lot of Pardalinum characteristics, but there's still some of that LA or Longiflorum characteristic in it.
Yeah.
Next slide. And Lancome, there's another one. Now, the Lancome bulb does not look like the bulb on the right, if I'm not mistaken. Is that correct? You species experts?
I don't, I
Langone
don't,
Gensei, you mean?
but Lankongensi itself,
Lankongensi, I'm sorry, yeah.
the bulb on the right, Lankongensi can look exactly like that.
Can it?
Can have
Okay.
a blush of pink, it can have a little pale part. So I wouldn't say, but I mean, you would have to have a lot of bulbs to see in that case.
Yeah. However,
Yeah.
this one really looks like a cross between an Easter lily and a species lily.
Oh yeah, and you can tell by the flower form. See how the
And the flower form really looks
the fragrance is
longiflorum.
different.
Yeah.
And
Yeah.
you can always see Lankongensi in the goosey buds. Look at the buds. Look at the shape. Look at the pedicels. They're always those goosey things with a goosey bud. You can spot longiflorum hybrids for pretty easily.
Yeah. So the bulb is reflecting that.
Yeah.
Okay, next slide. Now, Kushimaya is a cross between an Oriental and Nepalense, or a Nepalense cross and Oriental. When first dug out, the bulbs are whitish colored, look very Oriental, but as they age, they turn darker color. And you can see the flower again has a lot of characteristics of the Oriental and the foliage. So that's what's showing you again. We've got split characteristics in the bulb, but you have to look at the bulb very closely to tell whether you've got a true interdivision or not.
And sometimes you'll see that in the tissue cultures because the little bulb form of this tiny thing in the test tube will already be, you know, it'll be very like brownie eye. You'll see little speckles on the bulbs, and you'll see all kinds of traits at a very, very early stage. And then whether you grow the bulbs, the tissue culture tubes in the light or the dark will have an effect on whether it's purple or green
Or yeah,
or white. Yeah.
yeah, light does affect the color. Next slide. Easter morn. Here we have a longiflorum with a trumpet cross. And when they first dig out, well, these are both freshly dug out bulbs. One was Rick's and some are mine. At the bottom are mine. But if you look at this, you'll see a lot of the Easter lily look in the yellowish, but you'll see some of that reddishness coming out from the trumpet genes. And
Yeah,
the
look at
flower
the form.
form, the
Yeah.
flower form shows more of the longiflorum than it does the trumpet, so to speak. And look at the buds. They have those classic longiflorum type looking buds versus
Yeah, but the
trumpet.
inflorescence is not longiflorum. The inflorescence
No, well, I didn't say
is
that.
definitely trumpet. And look at the bulb's form. That's, you can see longiflorum in the bulb form.
Yeah, yeah, you can really see that in the bulb form. It's, yeah. So next slide. So no, now bulb color can indicate what the hybrid flower will look like. And here I've got two crosses. Both are OOT crosses. The one on the right, look at the color of the bulb. And the one on the left, and the one on the left, look at the color of the bulb. Now, they're both distinctly different colors. Now look at the flower form. The one on the left looks more oriental. The one on the right looks more Aurelian, and that's reflected in the bulb color.
It would really be interesting to know if those are triploids and they have two sets of trumpet genes in one and two sets of oriental genes in the other, you know, that,
Yeah,
because you can
yeah.
get it. But
Because I was quite
these are
surprised.
great pictures. Yeah, yeah.
I was quite surprised with the right that it was an OOT cross, because I looked at the flower and said, oh, what did I cross with that? But anyways, we'll see. I've hybridized with these two, so we'll see what happens afterwards. But yes, so the bulb color gives you an idea of sometimes what the flower will look like. Next slide. But sometimes the bulb color will differentiate an OT from an Aurelian. Here is Silk Road open pollinated. When I took it to the NALS show, the judge disqualified it because it said it was an Aurelian and not an OT. So I went back and dug up the bulb. And then the next time I took this flower to the show, I showed the picture of the bulb, because that is an OT bulb color. It's not an Aurelian. So yes, sometimes you think you've got an Aurelian, and if you look at the bulb, it'll be OT. And that's the
Well, and
class.
it's a question of how much as we keep breeding out over the generations. If you,
Yeah, yeah.
you know,
And there's the
little
other
bit-
thing, because the more you cross these OTs, the more sometimes you'll get more Aurelian looking, but the bulb
Yeah.
will show you whether it's an OT or just an Aurelian. A Henry I cross.
Yeah.
Okay, next slide. Then sometimes the flower looks one way, but the bulb says it differently. And this is one of my latest hybrids. It's got all kinds of Oriental in the background. But look at the flower form. It looks classic Henry I type. And
Yeah,
look at
not
the
just
look at the
form, but
color of the
pigment
bulb.
pattern, the pigment pattern, the papillae. Yeah, you see,
Yeah, yeah,
really interesting.
yeah. And so you look at the color of the bulb and you say, well, it is. And the background is OTs. It's just all OTs with some Oriental in it.
There's
Next
some
slide.
Henry I in there too.
There's Henry I in there too. Yes. Yeah. You know where the Henry I comes from? I'll show you. Next slide. One of the grandparents or great grandparents was Roy Select. That's what the flower looked like. And that's what the bulb looks
Oh,
like.
yeah, and that's where you get the flower form and you get
And the
the
flower
foliage.
form and everything
Foliage,
else. So
yeah, yeah.
obviously, that's probably the pod parent. But
And the long skinny scales, yeah.
and the long skinny scales, and that really, once it's out in the light, it'll turn, the whole bulb will turn dark, 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 dark. So yes, this was one of the grandparents or great grandparents of the previous one. And you see sometimes certain characteristics coming out in these hybrid crosses, and some genes will dominate. And in this case, it's the Henry I genes that will dominate. And because I don't do tissue culture and embryo rescue, selectively, I guess I've grown seeds that have Henry I in them because it's more immediate, hypogeo than delayed.
Look at
And
those Henry I pedicels. Yeah, you
Yeah.
really see.
yeah, it's yeah. Okay. And next slide. And that's it. Any questions or comments? Oh, there was a question down here. Where was this? Oh, we've asked that. Okay. No, I guess we've answered all the questions. Okay.
As did I want to say thank you, Rick, and thank you, Brian. You really did a great job.
And my thanks to
Absolutely
Rick.
Zero. I didn't even look at these pictures until this morning. I couldn't even drag myself upstairs. I've been
Yes.
sitting in the
Well, we're
living
so
room.
glad you're here.
We're still glad you're here.
Well,
Yes.
I'm going back to bed in a minute, I'll tell you, but
Okay.
I'm glad I was here, and I really appreciate what you did because this is
Thank you.
very helpful.
Okay. And Lynn, do we have any advertisements for